Okay, so now we've kind of finished up talking about the minute details and the mechanisms of the innate and the adaptive immune systems. We're going to kind of zoom out now and have a look at the broader effects of these systems on the body. So in this one, we're going to have a look at what happens when these immune systems are not working as well as they should be, in the case of immunodeficiency. And then in the next few videos, we're going to talk about what happens when they either also work too well in the case of autoimmune disease, and we're going to talk about when they kind of malfunction a bit and lead to resistance to harmless antigens that we call allergens for the hypersensitivity responses that we get when we are allergic to something. So let's just get into it on this one. For immunodeficiency, we're going to go over two examples of diseases for immunodeficiency in this one: DiGeorge syndrome and HIV. So there are two different types of immunodeficiency diseases. The first is congenital immunodeficiencies, which are caused by genetic abnormalities. So you're born with them. And then you have acquired immunodeficiencies, which are the loss of the proper function of the immune system due to various reasons like infection, nutritional abnormalities. If you're malnourished, your immune system isn't working as effectively as it should be, so you're kind of immunosuppressed, or medical treatments like chemotherapy. Abnormal components of the innate immune system in the case of congenital immunodeficiencies refer to defects in lymphocyte activation, maturation, and response. So congenital immunodeficiencies first. These are caused by genetic defects, typically found in X-linked genes. So they affect boys more frequently than girls because boys typically only have one version of these genes on their one X chromosome, whereas girls have two X chromosomes, so they're more likely to have the healthy gene on the other chromosome. Autosomal recessive defects, so genetic defects inherited on chromosomes 1 to 22, all the somatic chromosomes, so all the chromosomes that aren't the sex chromosomes X and Y, cause congenital immunodeficiency more frequently in populations with consanguineous mating. So that basically means congenital immunodeficiencies caused by mutations on chromosomes 1 to 22 are more likely to be found in populations that are high in inbreeding, because when you think about it, in order to have the immunodeficiency phenotype for chromosomes 1 to 22, because you have two homologous chromosomes for each of these chromosomes 1 to 22, in order to have the immunodeficiency, you need to have the gene on both of your chromosomes, both of your homologous chromosomes. And that is always more common in populations with more inbreeding, because inbreeding reduces genetic variation, so you're more likely to inherit the same copy of a gene on both chromosomes. So innate immune system defects may include non-functional enzymes, errors in leukocyte adhesion, or deficiencies in complement proteins. Whereas defects in the acquired immune system impair the functionality of the B and T cell responses. So that's kind of something to consider. So our first example for an immunodeficiency disease is DiGeorge syndrome, which is a congenital immunodeficiency disease. It causes a poorly developed thymus. So the thymus, as I'm sure you remember when we talked about the maturation of T cells, is the structure in front of the heart where antigen-presenting cells like dendritic cells bring antigens to the thymus, and then T cells with T cell receptors with an antigen recognition site that has a high specificity for that antigen are selected. So it's basically where the T cells kind of mature, and they also develop resistance to self-antigens here. The George syndrome also sees defects in the heart and thyroid, as well as a cleft in the upper lip that you can see in the images that I put to the right here, of the boy at the top and the girl at the bottom, both suffering from the George syndrome. The diagram at the bottom goes more into depth about the kind of physiological defects that you can see in facial structure, which is a good way of diagnosing the George syndrome. Moving on to acquired immunodeficiencies. These abnormalities are not of genetic origin, like in congenital immunodeficiency. Instead, they're acquired during life due to either infection, which we're going to go over in a minute with regards to HIV. It can be the result of cancers involving the bone marrow, like leukemia, because these cancers result in death of hematopoietic stem cells and the consequent myeloid and lymphoid progenitor cells, which give rise to all the cells of the innate and the adaptive immune system. So the bone marrow is very important to the immune system. If it gets damaged through cancer, then you're going to be immunosuppressed because you're not going to have as many of these immune cells being produced. And also immunosuppressive therapies like chemotherapy, which kind of damage these cells of the immune system. So human immunodeficiency virus, commonly referred to as HIV, is a retrovirus that infects cells of the immune system, mainly helper T cells, and it causes progressive destruction of these cells. HIV can be acquired through various means, including sexual intercourse, the sharing of needles, transplacental transfer, or infected blood transfusions. So, kind of ways of breaking through the barriers of the innate immune system, like the skin, to get to mucosal kind of gates of entry. So if HIV gets in the blood, or it's able to get through our dermis, through mucosal membranes in the genital regions, and then it can make its way to the blood through then, where it can start attacking these helper T cells in the blood. HIV infects host cells, integrates viral RNA into the host genome, and then proliferates and produces viral particles. AIDS develops over many years of latent HIV, progressively destroying the immune system, leaving the patient immunosuppressed. So there are 3 stages to HIV infection. The first is acute viremia, where non-specific fever-like symptoms manifest, which is symptomatic of initial HIV infection. This then progresses to clinical latency, where progressive destruction of CD4 helper T cells and lymphoid tissue slowly degrades the immune system. And then this develops into AIDS when the helper T cell count falls below 200 cells per millimeter cubed, and patients become susceptible to opportunistic infections, some cancers, and suffer from rapid weight loss caused by having a suppressed immune system. So they're very susceptible to infection at this stage of AIDS, which is how most people die. It can be treated by controlling replication of HIV and the infectious complications of the disease, so reducing exposure to pathogens. A scientist called He Jiankui used Cas9 to insert a premature stop codon into the CCR5 receptor gene in 2019, making embryos resistant to HIV infection, but this was illegal and he ended up going to prison for 3 years for illegal medical practice. If you look on the right, there's a boy called Ryan White.